Thank you. Let me add my welcome and thanks to amfAR for selecting San Francisco as the site of its new institute. We are really, really thrilled. So today I'd like to, I'm the guy with the blue glasses and the can of Roundup. That's me. Trying to kill those weeds, trying to kill the roots. So let me just, this is the problem. This is a picture of the fantastic progress and the problem. Before the mid-90s, we really had no effective therapy for HIV, and for individuals without therapy, their viral loads were very high, they progressed, and they invariably died. But then came along a combination antiretroviral therapy, and we were able, in most patients, to reduce the viral load to undetectable levels. Fantastic. We will go in the history of medicine, this will go down as one of the great milestones in all of medical history. It is a fantastic accomplishment. Now, we had hoped that those medicines would cure, but invariably, for no matter how well you're suppressed, no matter how long you're on the antiretroviral therapy, when the therapy is removed within almost 2 weeks, the virus bounces back. It comes out of hiding. It's coming from these latent reservoirs, these roots that we have got to get at. So what I'd like to, and the question is, well, is it possible? And I think that the field has been galvanized by the fact that, for example, Timothy Brown, the Berlin patient, has been cured. He's now 8 years out from his transplant with no detectable virus, off no medications. So yes, he went through 2 stem cell transplants and with a very special type of bone marrow transplant, but you know, it's an amazing story, and it gives us all hope that we can now operationalize such a cure and scale it. It has to be simple, it has to be safe, it has to be effective, and it's got to be scalable to the 36 million people in the world that are infected with HIV. What will our approach be in partnership with amfAR? Well, Rowena very elegantly introduced to you the concept of shock and kill. So these viruses, you know, you might say, well, why is it so difficult to cure HIV when I read in the paper that everybody with hepatitis C is getting cured with the modern medicines? The big difference with HIV is it takes up residence within our own DNA. It hides within our own chromosomes. So now the question is, how do we wake those cells up? If we can wake them up and get the virus replicating, we've got a chance of now killing that cell and removing the reservoir. So this is the typical shock and kill approach. Now, these are some shocking agents. We're not going to use these. We're going to use a different type of shocking agent that takes advantage of the innate immune response that Rowena also described, the toll-like receptors. But the goal is to get these cells to now being able to produce the virus. But the virus doesn't have anywhere to go because we're continuing the treatment with antiretroviral therapy. So we're blocking any new infections. But now this cell is immunologically visible. It will either be destroyed by our own immune system. It might, if we're lucky, just undergo a viral cytopathic effect and die on its own. We suspect, however, that we're going to have to not only have a good shock, but a good kill strategy to remove every last one of these reservoir cells. Now, we might not be able to get every last one of them, but we might be able to boost our own immune system to a point that we can control that, so that you can live with that very low level of virus and an immune response against it, the so-called functional cure. So those are the two. Timothy Brown may well be an eradication. He may well have had every last cell removed. We have to wait longer and longer and longer to see if he ever remits, but as of now, it looks like he is eradicated. That may be an aggressive goal. We may be looking more at tolerating a small amount of virus, but but with a good immune response against it. Now, we know that the kick, or the shock, can activate just about the viruses in the pink circle. Unfortunately, all of the viruses, all the replication competent viruses, the whole reservoir is represented by the purple. So we've got to have better kicking agents. We've got to have better strategies for really getting all of those reservoir cells activated. Because if we don't get them activated, well, we can't kill them. And it'll come right back and bite us in the rear as soon as we take away the antiretroviral therapy. So as Rowena mentioned, our strategy is to try and harness this more primitive part of our immune system, if you will, the innate immune response, the one that is the first line of defense, the one that springs into action in ours, to try and use it as the shocking agent and even as the killing agent. As I mentioned, we may not be able to get complete eradication. We may have to satisfy or settle for a decrease in the reservoir that allows the adaptive immune response to control the infection, thereby allowing discontinuation of antiretroviral therapy. Instead of kick and kill, perhaps what we would call reduce and control. So who's on our team? Who are the guys with the roundup? This is the motley crew. It's a wonderful group of scientists, some at Gladstone, UCSF, our IDRI partners, Blood Systems Research Institute, UCSF, the VA, Berkeley, and our good colleagues at Gilead. So this is the team that's really going to try and harness the toll-like receptors and turn them into great kicking agents. So let me tell you about toll-like receptors. So these are the pattern receptors that exist on the surface of cells that can detect whether a bacteria is in that neighborhood or whether something has entered the cell that shouldn't be there. The Nobel Prize was awarded in 2011 for the discovery of this family of receptors, the toll-like receptors, to Bruce Beutler and Jules Hoffman. So these, I mean, the fact that it was only awarded in 2011, these are not old, I mean, these are old receptors evolutionarily, but in terms of our understanding of them, they're pretty recent. This is what one of them looks like. It looks kind of like a weed, doesn't it? But it's a pattern recognition receptor, and in fact, the ligand is shown here in the blue and the red, sitting in the cleft and being recognized by this toll-like receptor. So we're looking now to use combinations of these toll-like receptors, not just one, but maybe 2 or 3, and to try and trigger them simultaneously to get a synergistic response. Now, is there any suggestion that that strategy might work? Well, here's toll-like receptor 4. It sits out here on the plasma membrane. Toll-like receptor 7 and 9 sit here inside in a vesicle inside the cell. So they're not even in the same places, but they're exerting their pattern recognition functions from different locations within the cell. So here's a TLR7 agonist having no response in terms of the production of this cytokine. We add a TLR4 agonist, no response, but we add the combination of a TLR4 and a TLR7, TLR4, TLR7, and sure enough, magic. This is the kind of synergistic response that we're looking to level at the latent virus inside a resting CD4 T cell. So can we use combinations of TLR4 and 7, TLR4 and 9, et cetera, to try and achieve this kind of kick or shock response? So the other question is, how are these toll-like receptor activators working? Are they actually acting right on the T cell? Well, the answer is probably no. Surprisingly, these TLR activators are probably acting on myeloid cells, macrophages and dendritic cells, inducing these cells to make cytokines that then activate the T cell. Okay, so if that's the case, by figuring out what the cytokines are that are being induced by these toll-like receptors, we might be able to get rid of the toll-like receptor agonists entirely and use the cytokines. And it might be a less toxic therapy. It might be something that it would be more manageable. So there's a huge amount of information that needs to be discovered in terms of how this cell talks to this cell in the presence of these activators. So, a brand new fast-track approach to discovering latency reversing agents. This is a very exciting area of science that's been really pioneered by Leora Weinberger at the Gladstone. And it turns out that HIV contains one of the noisiest promoters ever studied. Now, what do I mean by noisy? So noise is this biological process by which at any state, it can be it fluctuates from a higher state to a lower state. It's back and forth. It's never constant. It's always going up and down. And then, so the amplitude of the ups and the downs is the noise. And the LTR, the long terminal repeat of HIV, is incredibly noisy. Can we exploit that? So in fact, one can combine activators and noise enhancers, and together they can create this kind of magical response that now is able to activate the latent virus. Now the real key here is these noise enhancers would have been entirely missed in prior assays because they don't actually increase the magnitude of the response, just the variation around the constant mean. So no one would have ever discovered them. Many of these noise enhancers that Lior and his colleagues have discovered are already FDA-approved drugs. So we could be in the clinic with these noise enhancers in combination with other inducers quite quickly. And so being able to repurpose these drugs in quite a persuasive way. So that's going to be a very exciting and unique area of our amfAR effort as we try and use these already FDA-approved drugs in a different way. So one of the huge challenges in HIV cure research is that the latently infected cells are rare, roughly only 1 in 1 million. So think of about 1 million people, you know, maybe slightly more than the population of the city and county of San Francisco. And just say only one person in that city is latently infected. That's our challenge, is to find that one person, to root out that cell, get it activated without killing all the other cells. So is there any way to actually, and that cell, there's no way of recognizing it. It has an inert 9,000 base pair piece of DNA in its chromosome. Well, there wasn't any way to find those cells until now. Eric Verdin at the Gladstone has created these new viruses where we can now, the cells in red, the red cells here, are latently infected. We can find these cells with a trick of virology, and now we can purify. You can see that we could purify these cells. We could sort these. The green ones go here and the red ones go there. Suddenly we're now able to characterize and study the latently infected cell in a way that was never possible before. So we plan to use this technology along with our toll-like receptor to see how these cells respond to those toll-like receptor agonists. So we're trying a shock and kill approach, but shock without kill is a failed strategy. In fact, things could get worse. If you shock the cell and the cell begins to divide, the reservoir may get bigger. You've got to make sure those reservoir cells die. So how are we going to get those cells to die if they don't do it naturally? We're going to try and get these cells to die by one way, by using, as Rowena mentioned, antibodies that recognize, broadly neutralizing antibodies that are able to recognize those reservoir cells that have been shocked successfully. And we're going to use those antibodies to recruit natural killer cells, another component of the innate immune response. So combining adaptive and innate immune components to basically attack and kill that cell. Louis Lanier is an NK specialist and will be assisting in these types of studies. These types of antibodies are just now becoming available. Be a very exciting new approach for trying to kill the reactivated reservoir cells. But an area that I am particularly or very excited about is actually trying to use another component of the innate immune response to achieve the kill. So we're using the toll-like receptors for the shock. Can we use another component of the innate immune response to get the kill? And this is a part of the innate immune response called RIG-I. RIG-I recognizes RNA. Now, you don't need to know anything about this except that when a cell gets infected, the RIG-I with an RNA, if RNA is recognized, double-stranded RNA, by the RIG-I receptor, it launches an innate immune response that does two things. One, it makes interferon to protect all the cells in the neighborhood, and it induces the death of this particular cell so a two-part defense, cellular defense. Protect all the neighbors and die yourself. And so that's the question, this die yourself. So the idea here is to try and use a drug that induces Rig-I, acitretin, which is a drug that's already in the clinic being used for psoriasis. Zhou Wang and Leilin Ping are pioneering this the work, and the idea is to induce the Rig-I to a high level, and now in a virally reactivated cell where HIV is coming out of the reservoir, Rig-I might recognize that RNA, and the Rig-I might kill those cells that are preferentially being reactivated. And indeed, in tissue culture, these are patient cells from an HIV-infected patient. They can treat with acitretin, and they can actually preferentially deplete the HIV-infected cells, the ones that are the latent reservoir cells that are being reactivated. So it is a very promising and very exciting new approach to getting a kill that involves a second component of the innate immune response. So let me just leave you with the thought that, we're very excited about the prospects for the future, but it's quite clear that HIV research, cure research will likely be a rocky road, but the rockiest of roads lead to the highest of peaks. Thank you very much for your attention.